Head, the Department of Cellular and Molecular Biology, Madras Diabetes Research Foundation, Chennai. Over to Dr. Balasubramaniam.
Thank you, Dr. Jayanti. Let me thank the organizers, Shabu and Dr. Vijay Kumar, for giving me this opportunity. Incidentally, I saw that actually in the next hall, there are 4 people are talking about 4 different omics, technologies. Probably there might be a ectopic fat audience flow there. So I have a tough time of 20-20. I have 20 minutes. And let me see, this is the Lord Muruga and Lord Ganesha. In order to get the fruit, actually they have a different strategies. I don't know what to follow. So ultimately I should get some appreciation and get the fruit of appreciation. So this is my favorite slide. So as many speakers point out, there is a multiple pathogenesis in the type 2 diabetes, and Dr. D. Franjo called it as omnious acted. Dr. Madhu and Dr. Sridhar, they are all calling it as dirty dozen and treacherous 13. To me it is pathetic plenty, many mechanisms. If you look at the genes, the behavior, diet and nutrition, type 2 diabetes are very complex. So complex phenomenon always actually needs sophisticated solutions, and that's how actually Omics technologies, there is a lot of hype, a lot of hope too. So the rapid development of high throughput technologies and computational frameworks enables the examination of biological systems in unprecedented detail. The ability to study biological phenomenon at omics levels in time is expected to lead to significant advances in personalized and precision medicine. That's where a lot of actually interest to study these omics technologies. So basically the discovery of biomarkers should originate from genomics, transcriptomics, proteomics, metabolomics, and I add up actually 4 more omics also. So the genetic prediction of future of type 2 diabetes, in India a lot of work has been done. Dr. Mohan and one of my colleagues, Dr. Radha, has done a lot of polymorphisms. We also actually involved in Jiva studies. The combining the information from common type 2 diabetes with polymorphisms improves disease prediction. This is how everything started, and a lot of actually work has been done worldwide. But what you saw on the left, there are a lot of susceptibility loci for type 2 diabetes. It is a number of genes. It is 220 genes has been actually reported to be risk for type 2 diabetes. Even the right side, you can see that actually some of the genes actually identified in East Asians and ethnic specific South Asian kind of thing. So today, more than 150 genetic loci are associated with the development of monogenic, syndromic and multifactorial forms of type 2 diabetes. However, the proportion of overall type variants explained by these associated loci is only modest, 5 to 10%. This is a limitation. Effect sizes of type 2 diabetes associated genetic variants are very small. Odds ratio is 1:1 to 1:4 in the largely actually studied populations, studies like TCF7 like 2, and predictive value is not greater. That's a limitation. But what is a good thing? The genetic studies have implicated new loci in beta cell function. A lot of biology remains to be done to understand how these new genes affect beta cell function. Most of the pharmaceutical companies are sensitized by developing sensitizers. But now the focus is paradigm shift is on actually insulin secretagogues, more focusing on beta cell function. So that is the biology teaching. So jivas cannot read your future, but we have to go beyond jivas. If it comes to actually diabetes, you have to look beyond glucose. If it is diabetic retinopathy, you have to look beyond VEGF. If it is diabetic nephropathy, you have to look beyond microalbuminuria. Like that, we need to actually add on actually the other prediction markers. But let me talk about MODI. Some advantages occur. It's not a political MODI, it's a clinical MODI. Maturity onset diabetes in young. Here, you can now offer remedy for MODI. So while using the genetic information, you see here actually in the right, metformin doesn't have effect on these modifications. But the sulfonylurea, where the exact actually genetic defect is there, is correcting. So now it is possible to actually correct modifications with the correct precision medicine or personalized medicine. This is a genetic advantage. But in the other type 2 diabetes, it's very difficult, but at least in the monogenic forms in the neonatal diabetes, even if you can see that actually the neonatal diabetes switch insulin to oral drugs. Mutation in the potassium invertly rectifying channel subfamily member 11 gene represent another example in which genotype directs the approach to specific diabetes treatment decision making by the physicians. This is where actually genetics can speak at a clinical level. Then the proteomics, trying to hit moving target. The proteome is constantly changing. Technologically, you are always trying to hit a moving target. In certain aspects, you are looking at a dynamic system is growing and changing, and we can actually catch biology when it is happening. And because of that, we will find new mechanism and be more likely to develop new ways to look at mechanisms or affect them. Proteomics is also closer to drug discovery. But there are actually debates whether used to 2D or direct actually mass spec or something like that. The clinical proteome is getting a lot of interest. The good thing is actually all the bodily fluids can be amenable for proteomics. Whether it's a plasma, serum, tears, saliva, urine, vitreous, everything is amenable for investigation. And diabetic nephropathy, as I told, actually there is an unmet medical need. So we have to find actually the proteomics or protein markers before albumin appears in the urine. This is where actually a lot of work is going on. The urinary proteomics for early diagnosis in diabetic nephropathy, and there are two groups actually working in US to validate the 273 chronic kidney disease biomarkers in the type 2 diabetes cohort, and they are coming up with some kind of panel of proteomics biomarkers which can actually predict the development of diabetic nephropathy before albumin appears in the urine. And there are a lot of post-translation modifications. It's called PTMs. We all talk about actually insulin signaling, phosphorylation, all these things. We see here how much post-translation modifications. The list is actually enormous. We are talking about HDL is low, Asian Indian phenotype. Adiponectin is low. But what about the functionality? My HDL is actually 28 always. Okay, I'm not diabetic. So far I am a 54 and I'm okay. But maybe my HDL is functional. So we have to look at actually glutathione, albumin. The low levels are good. I mean, it gives actually some identification marker or something like that. But low levels of adiponectin, low levels of HDL, low levels of glutathione, albumin is not actually predictive marker. It is a function. So for that actually, we looked at the glutathione modification of hemoglobin in collaborating with the Indian Institute of Science, Professor Balram, and we saw that the glutathione-related form of hemoglobin modification is higher in diabetes and diabetes complication, particularly retinopathy. So this has been published in clinical biochemistry, and these are the studies actually tell that the power of post-translation modification of proteins, how they can be markers. And we are also actually working on advanced glycation where chemical biology meets medicine. We know that actually, despite the fact that DCCT studies showed that the independent of HbA1c, advanced glycation or advanced glycation end products are important. And I have given a recent talk, advanced glycation in diabetes, where too much advancement has disadvantages. And these kind of modifications are also getting a lot of interest. We are collaborating with the National Institute of the CSR Institute in Pune and developing some age markers also. Unfortunately, as per the proteomics is concerned, we don't have actually a large human proteomics project, although actually genomics has been done. And look at this human proteomic project, and the n number of actually the candidate actually the markers and all these kind of things. We really need actually a proteomics. Sometimes back actually DPT has formulated a clinical proteomics platform, but that has not been actually taken into practice, and we need a real proteomics, human proteomics platform in the context of diabetes. And epigenetics. This is another, recently it has been become increasingly clear that part of the gene environmental interactions relevant for complex diseases such as type 2 diabetes regulated by epigenetic mechanisms such as histone modification and DNA methylation. And microRNA is also coming there. And this is very important. DCCT study says like that, despite the actually prevailing HbA1c, the intensive group people continue to get protection. Whereas the other group, they got complication despite the HbA1c levels are actually maintained at 7.8 or the closeout period. That means there is an hyperglycemic memory that is now explained by epigenetic modifications. And there are a lot of studies coming up epigenetics, and again in India actually epigenetics is Dr. Yajnik, and actually one or two people are actually working on it, and we are also started working on histone deacetylases, and this is an important area. We need a lot of contribution in this area. The reason is actually whatever you see actually the risk factors for diabetes is somehow actually controlled by epigenetic modification, and your gene function, gene expression is based on the epigenetic modifications. And newer drug target, pharmaceutical companies are getting a lot of interest. You can develop histone deacetylases, you can develop histone acetyltransferases, you can develop sirtuin activators, histone methyltransferases, histone demethylases. One of my students working on HDAC, and we found that some isoform of HDAC is very, very highly actually represented in type 2 diabetes, and we are developing actually some inhibitors of HDAC inhibitors in the integrated institute of the Jammu, the CSR institute. We got some natural products which can act as HDAC inhibitors in the context of diabetes, and we need to work on that more. And microRNAs, and this is actually getting a lot of interest. Your mRNA can be not stable, protein cannot be stable, but unfortunately or fortunately microRNA is stable. You can use all the actually body fluids to get some kind of microRNA information because they are stable, they have tissue specificity, they represent pathophysiology and physiology, they have a big and robust discriminative power. If you have actually disease and we have a normal actually patients, you can have some microRNA discrimination. So this is the some of the studies actually getting a lot of interest because we can study the tissue microRNAs or also body fluid microRNAs, serum plasma, and Satinath also told about inflammatory component. Yes, there is a strong inflammatory insulin resistance component in Indians. What we did is, whenever actually inflammation increases, enough kappa B increases. When enough kappa B increases, there should be induction of a microRNA called 146A. So we looked at and we saw that that is downregulated in diabetes, and it has correlated with a lot of the target genes, and we have showed that actually underneath the impaired 146A there is more inflammation, and we have published this in the molecular and cellular biochemistry. Impaired VIR146 expression links to subclinical inflammation, insulin resistance, and type 2 diabetes. It is also carried in editorial in Nature India, and that's going on. And we also, at the first time in the country, we have taken vastus lateralis muscle cell. So where is the main problem, glucose uptake problem? It's in skeletal muscle. So we have taken the vastus lateralis muscle cells from the control, from the pre-diabetes, and then the diabetes people, and we use the microRNA technology, and we are getting, already we got some information, and just I am processing it for publication. So it already showed that actually there is a distinctive RNA signatures in the pre-diabetes and diabetes in the skeletal muscle. And we also actually using the circulatory microRNAs. I got a DPT project to look at actually what is the difference of microRNAs in the serum from control, pre-diabetes and diabetes, and it is also giving some very good actually South Indian phenotype specific microRNAs. One microRNA which has not been reported in 7 publications so far in the world literature, and this is also getting ready for the publication. And we are also talking about actually why lean diabetes could be metabolically obese, and we saw that actually even in the lean diabetics, adipocytes are hypertrophic. They are senescent, their telomeres are shortened, they release actually more inflammatory markers. they release less adiponectin. So the first speaker actually, the mother actually, told that the sick adipocytes. That's exactly what we are also seeing. Subcutaneous fat is also important, but subcutaneous fat is also actually sick. We have looked at actually the microRNA profile, and that paper is also getting ready, and we saw that actually visceral fat and both subcutaneous fat, the quality of the fat is matters, and they are sick. And there are specific microRNAs that are currently being pursued in clinical candidates. We have to go a long way. And metabolomics. And metabolomics is considered to be the best indicator of organism phenotype. So when you compare genomics, proteomics, and transcriptomics, metabolomics are actually the end products, and we can see them as a biomarker. So we can have a targeted metabolomics or untargeted metabolomics. The targeted metabolomics, we can have a biomarker quantification, whereas untargeted metabolomics can have actually a discovery biology. So we may get ready for an acid test for diabetes, because there is a Nature Medicine paper just looking at 5 amino acids: isoleucine, leucine, phenylalanine, valine, and the other which is called the tyrosine. They discriminate who will get actually diabetes in the Framingham study with a 12-year follow-up. So there might be an amino acid test is also possible, and the same Framingham study, the 2-amino adipic acid has been actually evaluated as a biomarker for diabetes. So this is getting a lot of interest, and there is one more actually glyoxylate and other mechanisms also metabolic study has shown. The interesting thing is actually by these metabolic studies we can get the risk for diabetes, and also who will get benefit from calorie restriction. Who will get benefit from bariatric surgery? Who will get benefit from the loss of weight, exercise? All this information is possible through the metabolomic studies. And one more layer of complexity, metagenomics. This has been covered by Dr. Ganapati. Your diabetes fate is not only decided by your own genes, but also by your gut microbial genome. Unfortunately, we don't have actually Indian data. There are 7 studies all over the world on metagenomics. Metagenomics is actually important because all the medical microbiology techniques available worldwide can identify only 20 to 30% of the microbes. That is the limitation. By using the actually cysteine RNA sequencing, you can catalog all the trillions of gut microbiota. They are present in oral or actually in the gut microbiota, the intestine or something like that. So a lot of interest is happening, and Dr. Ganapati has covered all these things. Unfortunately, actually, Dr. Mohan and colleagues and myself, I am also an investigator. We got a big Indo-Danish grant to look at the microbial diversity in the type 2 diabetes patients compared to pre-diabetes and diabetes that's going on in collaboration with the THSTA in New Delhi and the bioinformatics support given by TCS. So we have done another study, the LPS lipopolysaccharide has been actually shown increase in diabetes, and we have provided the mechanisms also. This is the first study clinically shown that LPS and the zonulin are increased in type 2 diabetes. And I have also wrote an editorial, Metagenomics Health Claim. Are you rich enough in your gut microbiota? This is getting a lot of interest because better health may not be directly related to how rich you are financially, but it is going to be related to how rich you are in our gut microbiota. This is getting a lot of interest, and a lot of metagenomic studies are needed country level. And pharmacogenomics, metformin is actually thing. All this metformin. I wrote an article. Nature gives that keeps on giving more. But metformin actually some of the patients they don't respond. The reason is actually there is an organic cation transporter with which actually the metformin uptake will be reduced. And similarly, there is a multi-drug toxin extrusion protein where actually metformin uptake can be more also. So these kind of pharmacogenomic studies are actually very much important to know, to make the decision by the clinicians. And there is also a lot of actually concept on nutrigenomics, using that nutrition and other things for not only the genomics point of view, the epigenetics point of view, where actually DNA methylation, histone modification and microorganisms can be actually beneficially altered by diet and nutrition. And another actually complexity is human toxome project is also coming up. This toxomics. there is actually role of endocrine disruptor, and we just started actually looking at the endocrine disruptors, including bisphenol. The World Health Day just gone actually 7th April. And food safety, food is plenty, but what about the food safety? And these are the environmental contaminants are also actually connected to type 2 diabetes. We need to study them, this toxomics point of view. So technology is still struggling to unravel the guardian knot diabetes pathogenesis. The task or challenge before us is how to integrate these concerted efforts of Omics technologies advancements into a holistic and translation solution so that there will be a real value addition in clinical situation both for diabetes prevention as well as better diagnosis and treatment. There is also actually, there is a Stanford, there is a technology is getting developed. This is called IPOP, Integrated Personal Omics Profiling. This is a 3.6-year study. 14-month study has been over. Longitudinal profiling of all this personal omics profiling using the genomics, transcriptomics and antibody omics and the metabolomic studies, and they have shown that within the 14 months time the onset of type 2 diabetes related metabolic signatures have been identified. There is a long way to go, but this integrated personal omics profiling is what actually people are looking at in the long run. So we need to transform data to meaningful clinical decision making. That's the big challenge. We need building borderless minds and borderless thinking. So everything is actually very important at the concept of actually the basic scientist, the computer modeling person, and whatever that algorithms we are going to make, what kind of actually additional benefit we are going to get in terms of actually decision making in treating the patients, preventing the diabetes and also prognosis point of view. So the last slide is actually, the one good thing is lifestyle changes matter. Everybody says, even a molecular biologist like me, I am saying the same thing. It can prevent diabetes. All the omics advancements certify health benefits of exercise. I wrote an article, TCF like 7-2 diabetes, a transition gene with a prescription hope. That means the 5 days actually exercise, the risk for gene variant of TCF7-like 2 is negligible after 6 months of lifestyle modification. Metagenomics, it is supporting actually exercise and lifestyle modification helps. And metabolomic studies also says the same thing. And microRNA studies also saying that actually lifestyle modification can actually do a beneficial gene expression changes. That means I tried all the fitness pads, but my doctor was right all along. Walking is still the best exercise. So in order to actually promote, whatever that Vishwamitra, Vasishtha has to actually certify him as a Brahmarshi. So as a molecular biologist, we are giving all this omics actually advancement to say that actually lifestyle modification is important for not only diabetes, for all other non-communicable diseases also. So India has an advantage. We have diverse clinical phenotypes, physician scientists, higher education. UNESCO has just today mentioned that actually India quality of education is improving. Infrastructure, IT, modeling, everything is there, and we have a skilled human resource. So Modi says make in India, and I say make in Chennai also, because Chennai has contributed a lot in the field of diabetes. I thank Dr. Mohan and my disease biology team. Thank you.